It's quite a pleasure to be here and tell you about the work that's been going on in my group in two-dimensional high-throughput microscopy screens. And the data visualization that we do is really just beginning. So I think the purpose of my talk is partly to convey the enthusiasm that there is in the biological community for this type of experimentation, and also the challenges that are in this field for data visualization, because there are many, and we could really use some help. So in my talk today, I will be talking about 2D microscopy images. It turns out visualizing them is very simple. I just did it. The problem is if you have a million of them, how do you visualize them? You don't want to look at them all by eye, and so it actually becomes more a problem of visualizing image data. So each image that we acquire by microscopy has to be turned into some set of numbers that is conveying some sort of biological meaning, and that's where the real challenges come in. So it is not surprising that our field is somewhat lacking in visual analysis tools, because we have, in each image, typically 1,000 cells. Each cell, we can make 1,000 or so measurements of all of its traits, its colors and shapes and the textures of the different stains that have been used for the cells. And then we have, we usually have 100,000 or maybe even 1 million samples to process. And so the explosion of that amount of data is really where the challenge lies, and that's what I'll be talking about today. So in our typical experiment, we have a large number of multi-well plates. Each one has some cells in it, or in some cases organisms. We also do some experiments with C. elegans worms or maybe zebrafish. Each cell sample is treated with either a genetic or chemical perturbant of some sort, something that does something to the sample. And our goal is typically to find which samples are interesting, which ones have an interesting phenotype, phenotype being a property that's related to the biological process of interest or the disease of interest, and so on. And so with these multi-well plates, you feed them to an automated microscope. It's a robot microscope. Many of these are commercially available at this time, and collects thousands to millions of images. You process those images using some kind of image processing software, and that generates cell measurements. And as I said, typically it can be hundreds or even 1,000 or so different features can be measured for each individual cell. As you can see, you can identify different compartments of cells from the images. And this step is becoming more and more straightforward. For most cell samples, it's actually not too challenging to do this step. Of course, there are a lot of things we're still working on in this area, but for the typical human cell type, it's not too challenging. And then lastly, what I really want to focus on today is the data exploration that has to happen downstream. Basically, how do we turn that gigantic database worth of numbers into something biologically meaningful? And I'll note that in our own laboratory, we've generated, as Thomas mentioned, the open source software called Cell Profiler, which is widely used for this step, turning the images into a large set of numbers. And then we also have a tool which has just a few of the things that are feasible in this field, but there's a lot still to be done. And so this is much less well developed, and where we're really excited to do more work. So the team, I want to make sure I emphasize that the work I'm describing today is largely the work of myself and Ray Jones from our PhD and postdoc experiences, but now we have a group with about 10 people, and they contribute very heavily, along with all the biology collaborators. So these experiments, what are the biologists actually doing in each experiment? The goal is to look for samples with some unusual phenotype, some unusual property, and the samples that we're testing are typically genetic, so RNA interference to knock down every gene, or maybe overexpress every gene, or treating with small molecules, chemical compounds that we're hoping to find perhaps a treatment for a disease or a perturbant of a particular biological pathway. So these experiments are tremendously useful to identify genes that are involved in biological processes, to identify chemical probes that we can use to study processes, to identify drugs, to treat diseases, and as I'll talk about, we are actually starting to use these experiments to identify biological processes we didn't know existed or certainly didn't understand well. So I'll start with a very, very simple example as far as how the images look. In an experiment that we're working on searching for tuberculosis treatments, it's conceptually very simple. The blue is human cells, actually nuclei, and the green is this tuberculosis bacteria that's been labeled. And in the absence of drug, you put the two together, the cells end up infected and pretty unhappy and have a lot of green blobs on them. If you treat them with a successful antibiotic, that infection does not take place and you don't see green blobs. And so this is, from an image analysis perspective, relatively straightforward. You identify the cells and define compartments for each one of them. You identify the bacterial blobs, and then in some fashion you count up the number of bacteria per cell. Okay, so that's pretty straightforward, and I won't spend much time talking about that. Well, actually, I won't spend any more time talking about that. But where it becomes more interesting is when you have more challenging phenotypes. So here's a set of 14 phenotypes that we wanted to score from images, but it's not so simple as counting the number of green blobs. We wanted to characterize these samples for these different phenotypes. Some of them are linked to diseases. For example, this is a colon cancer cell line that we're testing here, and these protrusions are known to be related to certain types of cancers, as are the crescent-shaped nuclei, which are shown in this panel here. So we wanted to score these multiple different phenotypes. These have kind of more red staining at the edges, and they're more flat and omelet looking. But we could not come up with such a simple measure as number of green blobs. And so this is one of the challenges that we're facing. And so I want to cover 3 points in the talk today, data visualization challenges that are remaining in this field. Number one, how do we assign each sample a numerical score? In the case on the left, it's quite straightforward. In the case on the right, it's a bit more challenging. Secondly, how can we explore these scored samples once we've assigned them a score? How do we narrow them down to a small number of hit samples that we want to study further? Usually the biologists have limited resources to follow up. So if they've tested 100,000 chemicals, there might be thousands of them that have some amount of the property that they're looking for, maybe some efficacy curing tuberculosis, for example. But how do we narrow it down to just a small number that is feasible to follow up on in the laboratory? And then lastly, how can we extract more information from these experiments beyond just a list of the top 10 chemicals or the top 10 genes for any one process? So we'll start here with the how do we assign each sample a numerical score when it's a complicated phenotype, when it's not terribly easy to find a single measurement. And the answer that we've followed pretty successfully is to just measure everything you possibly can about the cell and then ask questions later. So if you take an individual cell from one of the samples in the experiment, cell number 6 million, blah, 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 you want to measure every one of its properties. You know, how big is this blue compartment? How big is the red compartment? How is the distribution of the red relative to the blue, and so on. It turns out you can measure hundreds of features, and I kind of zoom in to show you some examples of those features here. And we call this strip of data here a cytological profile. It's a collection of measurements that describes the appearance of the cell numerically. And so we have that vector for each cell in the entire experiment. So if you look at it, this is just showing 4 different samples, because more than that would be overwhelming. So here's 4 different samples. It doesn't matter what they are, but each of them has a certain number of cells, maybe a few hundred of cells, and then several hundred features that have been measured. And you can see there's patterns that, you know, this sample here seems to have cells that mostly share a good proportion of features. A lot of the features are correlated with each other. You can see some interesting patterns in this data. Now, why do we measure everything? Well, it turns out, I won't go into the details, but machine learning is a very useful way of identifying cells that have those more challenging phenotypes that I described. And so I'm guessing many of you know what machine learning is, in which case I won't explain it to you. Many of you don't necessarily understand how it works, in which case I'm not going to explain it to you today. But I'll just show you what it looks like. So that shows this in action. So this is the tool that we built for machine learning. And it's designed for biologists. And basically, cells from the experiment are fetched up here. And the biologist is picking those that have the phenotype of interest. In this case, he is picking some cells that have kind of this green staining, just to make it a very simple example. You sort a bunch of cells, and you can see that if we zoom in here, let me stop the madness here and go back. What you can see is over time, as the biologist is sorting the cells, you start to ask the computer, show me more cells that look like the cells that I'm looking for, okay? And so we use gentle boosting as a form of the form of machine learning that we use, but in reality, just about any kind of machine learning method works well here. And what this allows you to do is very quickly, usually within the course of a few hours, sort cells and get the computer to learn the phenotype straight from the biologist. And so having this tool has been really helpful to score quite a number of phenotypes, and I'll show some examples of those. So aside from these 14 that I described before, we've been working on another dozen or so projects, just 2 of them I show here. In one case, we're distinguishing between neurospheres and adherent cells, looking for genetic regulators of glioblastoma, which is a largely untreatable form of brain cancer. And also looking for genes and chemicals that can regulate whether cells are leukemic or just hematopoietic and non-harmful to the human body. And so being able to quantify these really complicated phenotypes in a straightforward way, which allows the biologist direct access to defining the phenotype, has been really useful for us. In other laboratories, just to give a broader overview, the Murphy Lab, for example, at Carnegie Mellon has been using machine learning methods to score things like localization patterns. So in this particular cell type, they have many different proteins labeled with this green dye here. And what it's showing is that he's shown that using machine learning methods, the computer can distinguish these different patterns of localization and assign each cell a score for what proportion of the green protein or the green dye is of each particular known pattern. So I think number 1 is typically pretty well handled right now, as I mentioned, especially with the machine learning methods that have come out more recently and the usable tools for it. So this brings us to the second data visualization challenge, and it gets progressively more difficult as we go. How can we explore the scored samples to narrow down just to a small number of hits that the biologist can actually study? So first, there's some quality control steps. First of all, do my controls look good? And so if I have 2 replicates of an experiment, all we need really is a scatter plot. There's 1 data point for every image. Each image is scored for whatever the phenotype is. And you want to see that your controls are pretty tight, that there's not too many outliers. And typically, we just turn this into a single metric of assay quality that defines the standard deviations here and the difference in the means between the 2 populations. And that gives us a sense of whether the experiment has worked well. So that's pretty straightforward. There's things we can do looking for systematic artifacts. We might want to look at the experiment in the layout that it was actually done. So each of these blocks represents a multi-well plate in the format it was done. And there's a well-known effect where typically the way that the wells evaporate, because there's liquid in each of these wells with cells growing in them, typically the evaporation that occurs at the edge of the wells, at the edge of the plate, can cause problems with the phenotype being scored properly. And you also have other strange effects, like something happened at these rows, or something is going on with the pins that are trying to add cells to the samples, and so on. And so there's been tools developed. These are from Acuity Express, which allows you to browse and correct for these systematic artifacts. You can also look for maybe a bit more sophisticated, looking for, is the phenotype that I'm interested in confounded by other factors? So let's say, for example, I have a phenotype of interest, and that phenotype of interest actually is correlated with something like the number of cells in the image. So in other words, any treatment that I do to the cells that decreases and makes them be more sparse is going to kind of look like my phenotype of interest. In a case like this, I may look at a plot like this, and it helps me to make the decision that I'm actually only interested in these 2 orange dots down here. Those are the only 2 samples where I see the phenotype of interest, but I have a relatively normal number of cells in the image. So those are the kinds of decisions that people may need to make in order to decide how properly to score their samples. And once you have a nicely normalized set of scores for all of your samples, you may have hundreds of thousands or millions of them, you might look at that data and choose a threshold that defines your hits versus not hits, and you might explore the properties of those. Where do they fall amongst the plates in your screen? Did all of your hits happen to come from Tuesday instead of Thursday, in which case you might want to go back and recheck your data and do further quality control, and so on. And this is a screenshot from a very nice tool, Spotfire, which is widely used in the field to explore this type of data, and has a lot of really nice visualizations in it. And once you feel that you have good hits, the end point of every screen is a biologist sitting down looking at the images by eye, just making absolutely sure that the hits that they plan to follow up on are really real, so that they look like this instead of like that, which is unfortunately not all that uncommon. So that concludes our how do we narrow down these, doing quality control and really identifying the hits. But what I think where the field is most stuck at the moment is how can we extract more information from these experiments? If our goal is not just to say what are the top 10 chemical compounds in my screen, but instead to say there's a lot of information in each of those images. Every image has 1,000 cells, and every cell has 1,000 properties. And by perturbing them with a million different chemical compounds, we have the potential to gain quite a lot of information aside from just what are the top 10 chemicals that kill tuberculosis, for example. So one thing we can do once we have the hits from a screen is explore those hits relative to, or explore the phenotypic score relative to other sources of data. And so here, the source of data is I have increasing doses. And this, again, is from the Spotfire tool, where for all my different this small set of samples here, I can look and see whether the phenotype is correlating with the dosage of each of these different chemicals that have been tested and kind of confirm that that's dose relevant. I can explore other properties of the hits. This is a tool, an open source tool called KNIME, which is based on a pipelining kind of program. It's a Pipeline Pilot is a commercial software that uses a similar concept of visual programming, where the biologist places a node that has certain properties and certain nodes can connect to other types of nodes and so on. And this KNIME tends to be very good at visualization. And so, anyway, you can see the different typical types of plots. And if you're kind of interested in certain sets of samples, you might say, I want to see all the samples that are my positive controls, and where do they fall along these parallel coordinates or in a particular scatter plot and so on. So exploring those properties can be very helpful. And what we'll hear later in this session, I won't spend time here since it's not something that we focus on, is clustering. Can we identify within this large data set, let's pretend we don't know what phenotype we're looking for. Are there things that are happening to these cells that might be of interest to us? So in an exploratory way, if I'm working with a glioblastoma cell line and I think I'm looking for a particular phenotype, is there something else there in the data that I should pay attention to? And so one method of coming at that question is to cluster individual cells and try to identify whether there are any unusual phenotypes that maybe I wasn't expecting to see in the experiment. Typically, this is done by, historically is done by a biologist just flipping through images and making sure there isn't anything interesting and unusual. But the ability to do this computationally is really helpful. And so you'll hear a talk about that later in the session. In particular, what I find most fascinating is the idea that can we allow the algorithms to tell us that there's a particular type of cell that is unique and is measurably different from the others, but may not even be visible, that difference may not be visible to our own eye. So it's visible in the sense that it's captured in an image, but not necessarily visible to the human eye. There's certainly many properties of cells that the computer can capture that we just are not tuned to look for. And so I think that's a very exciting aspect of using algorithms to extract features. Another approach for identifying things you may not have expected in an experiment is to do something like self-organizing maps of whole samples. So this is an example from Acuity Express, another type of software that's common in this field, where each of these blocks represents a dose-response curve. So the left is a low dose of the compound that's been tested, and the right is a higher dose. And the idea is that all the data from a certain number of samples here has been fed into the self-organizing map, and then it's positioned everything near each other such that similar samples are near others. And so if you happen to know that this sort of everything here is kind of typical, this is where your negative controls lie, maybe this is the phenotype you were looking for in your screen, but by looking at this kind of visualization, you may notice, hey, there's this other group of samples that has something I maybe wasn't expecting, and then that would be the basis for further exploration. And then lastly, what we're very excited about is looking for patterns and similarities and relationships across every axis. So we have this gigantic data set. thousands of features, perhaps across entirely different assays. So if we use the same RNA or genetic or chemical perturbations on this axis, and we test those across many different assays, and each one has a few thousand features that have been measured, then we have this very large data set. Historically, what I focused on and what the practical outcome of this field is so far is answering the simple questions: what genes are involved in my phenotype? What chemicals affect my phenotype? Which is typically answered by just picking one column of this gigantic matrix and picking the top 10 chemicals that meet that criterion in that one column. But because all that information is there, we can also answer questions like, irrespective of the assay, irrespective of the features that have been measured, what genes that I've tested just appear to be similar to each other across all these different assays or features? What chemicals are similar to each other? And perhaps even more excitingly, if you've tested both genes and chemicals in the same sorts of experiments, you could ask the question, what genes look similar to a chemical? Because this would indicate the chemical's potential target, that the chemical is targeting a particular protein, and that's what's yielding the effect that you see in the cells. And that can be very useful if you are headed down the pharmaceutical path to know what the target is of a particular drug. You can also say, I don't even care what the chemical or genetic perturbations are. I'm curious what phenotypes are coordinated to each other. You may notice something in the data, for example, that every time the nucleus has a funny wiggly-edged shape, it turns out that some reporter protein from some other assay is always elevated. And so seeing a coordination like that leads to a hypothesis that is testable and potentially quite interesting. And then these were all answered within a set of image-based experiments, but we can also, of course, look at what relationships exist between these phenotypes and known information about the samples from external sources, so protein-protein interactions, transcriptional microarrays, and so on. So there's a lot to be done here, and a lot of useful things that can be done. I mean, typically what's done is clustering, and this is often qualitative. The typical approach is cluster your hits, put that as figure 5 in your paper, find something that makes sense and comment on it, and you're done. And I think the field could advance more from that point. This just shows, again, just clustering. Here we have dose response data, so you can kind of see whether the phenotype is increasing across doses, and how does that phenotype compare with other phenotypes that are seen in a set, and so on. But we really don't have a lot of tools in this area, and so I'm excited to have more arising. So that's all I wanted to talk about today. Just wanted to mention that at the Broad Institute, it's a nonprofit biomedical research organization, and we have quite a large group of people who are dedicated to screening chemicals and genes. And my own group, the imaging platform, is available to work on image analysis projects as well. So with that, I would like to thank you and be happy to answer questions.
There's a long history of microanatomists looking at cells and defining what they thought those phenotypes were, rather than just picking them. How well do the machine learning ones correlate with what's in the literature and the sort of traditional ways of phenotype components?
Yeah, that's a great question. We found that we've done all kinds of different quantitation for all kinds of different phenotypes and so on, but in general, it turns out that there's just as much discrepancy between two biologists as there are between the biologist and a computer, is probably the simplest way to explain.
Is that a large number or a small number?
Large number or small number?
The discrepancies.
Right? Oh, it's a pretty large number. It's a pretty large number, and I guess, it depends on your, any phenotype can be, so usually a phenotype is defined in the biologist's mind as yes or no, but any two biologists have the threshold slightly wherever it's going to be, and the computer would be the same way. It tries to learn where that threshold is from examples, but typically we as biologists are not that consistent about where we place thresholds in our mind, and so it's a bit of a complicated thing to approach, but in general I would say that the computer does as well as human beings, which is in some cases, 85%. It's certainly not 99% consistency.
Sorry, this is after training or actually for the clustering approach you are referring to? This is after training.
Assigning a score to a phenotype is a sort of inconsistent thing across biologists.
Okay, so then we could just discuss the same thing for clustering, actually. So if you just compare the results of clustering different nuclei or cells with a human expert, do you get then similar? Do you reach a similar conclusion, or then would you say, okay, then this is really completely different?
So we haven't done clustering experiments on individual cells to any large degree. So I'll leave that to Nick and Mark to talk later.
Okay. Yeah. Okay. How can a computer manage to quantify things you can't really see as a human expert?
So it's actually not that mysterious, because the human brain is not particular. There's certain optical illusions and things that we're just not tuned to identify. So for example, if you look at 100 cells of this type and 100 cells of this type, and one is maybe 10% larger than the other, if every single cell is 10% larger, you may not notice that by eye, but the computer can very easily detect that. Even if it's 2%, if it's consistently 2% larger, it can detect that. Maybe something that's slightly more interesting, but still pretty straightforward, is the blue staining that we tend to use for nuclei, the DNA stain, is a quantitative stain, and when the cell goes through the cell cycle, it doubles its DNA content. The human eye is very bad at detecting just the bulk amount of intensity coming from a single cell. And so the computer is very good at telling whether a cell has one-fold or two-fold amounts of DNA, but human beings just aren't very good at two-fold levels of bulk intensity coming from cells. So it's another not so mysterious thing that the computer just happens to be good at. Other things would be certain types of texture measures. So there may be a texture measure that says, you know, for every pixel that's bright here, how many pixels five distances away are also bright. And that's something that we may or may not be able to detect depending on how it actually looks. So there's quite a lot that the computer can do that we're just not really designed for.
So the question was if this can then be double-checked, and if you double-check it.
So the computer can draw our attention to things which then sometimes we can see. And that certainly happened to us in several cases. So then it's funny because we spend so much time having us as biologists teach the computer, but then the computer teaches us what it is that we're actually looking for. So to summarize Nick's answer there, he's just pointing out that the human eye can only distinguish about 30 levels of intensity gray levels, whereas computers can look at many logs of grayscale intensities. And there is the possibility to adjust color maps so that the human eye can see particular gradations if you know what you're looking for.
Okay, is there one more? Okay, this is the last one. The question is about the redundancy of the 100 parameters, or, well, n100 parameters you're actually measuring for your cytoprofile.
Yeah, so you could get a bit of a glimpse of that just from the data that I showed, that kind of, you saw many columns seem to track in line with each other. And so certainly, so when we measure 1,000 features of each cell, there are a good proportion of them that are redundant. The machine learning methods that we do are pretty resistant to redundancy. So the gentle boosting, as an example, picks the best feature, and then given the best feature, picks the next best. So the second best feature is certainly not going to be one that's highly redundant with the first. And so we try to train the algorithms to handle the fact that there's perhaps very many correlated features in the data.
And it is particularly tricky to define redundancy. So that's, so with this, thank you very much.